Good evening, students. Good evening. Whether I'm audible? Good evening, everyone. Yes, whether I'm audible? Yes, good evening. Yes. Yes, today we are going to see one of the most important topic. Okay, today we are going to see the metabolism of major biomolecules. Okay, we are going to see metabolism of carbohydrates, proteins, lipids, and DNA molecules. Okay, we are going to see everything today. Okay, if you listen this class, you don't want to go on reading Leninger book or instant notes on biochemistry. These notes are highly sufficient to cover the biochemistry metabolism portions. Okay. So if you have any doubt, kindly let me know there itself. Okay. First, we are going to start with fatty acid metabolism. Okay. From every year, if you see the question paper, you will get minimum 2 to 3 questions from fatty acid metabolism. Okay. First, we will see how the fatty acids are synthesized. How the fatty acid are synthesized. Then from fatty acids, how these fatty acids are utilized for synthesis of membrane lipids. At last, we will see how the cholesterol molecules are synthesized. Okay. Then we will see how the catabolism of lipids will happen. This is first part of our class today. Okay. First, we will see the general definition of what is meant by lipids. Okay. Lipids are generally hydrophobic or amphipathic biomolecules. Okay. These are hydrophobic molecules. That means it will be highly soluble only in organic solvents like ether and ethanol, but insoluble in water. Okay. Some of the lipids may be amphipathic. That means it will have both polar and non-polar groups will be there. Okay. Then if you see the major functions of lipids, this is one of the important questions. If you see the major functions of lipid, it will act as a storage molecule. For example, triacyl glycerides will act as a storage molecule. Okay. Apart from that, these lipids are involved in structural component of membranes. Okay. Lipid bilayer will be made up of majorly of lipids and proteins. Okay. Then some of the lipids may also act as a signaling molecules that we are going to see in our lecture today. Signaling molecules. Okay. Then some will also act as a precursor of hormones. In yesterday lectures, we have seen lipid alpha linolenic acid. This fatty acid will act as a precursor for jasmonic acid. Like that, this fatty acid may also act as a precursor for hormones as well as for vitamins. For example, A, D, E, K. These vitamins are also having this lipids. Okay. The major classes of lipids, if you see, first fatty acids, then triacyl glycerol, which is a storage biomolecule. Apart from that, lipids, phospholipids will be there, sphingophospholipid and sphingolipids will be there, then glycolipids, at last sterols will be there. These are the major classes of lipids. Okay. First, we will going to see how the fatty acid molecules are synthesized in plant as well as in mammals. Okay. The question what they will ask in fatty acid synthesis, first I will tell. Okay. Where the fatty acid synthesis will happen. The key difference is in plants, the fatty acid biosynthesis will always happens in plastids. Okay. This is most important thing. Okay. In plants, the fatty acid biosynthesis will be happen in plastids or chloroplasts. But in mammals, cytoplasm is the major site for fatty acid biosynthesis. Okay. Then we are going to see how the fatty acids are synthesized. Okay. The major recruitment for fatty acid synthesis is first is acetyl-CoA molecules. This will be condensed. This acetyl-CoA molecule is a 2-carbon compound. This will be condensed to form the fatty acids. Okay. For example, 4 carbon, 6 carbon, 8 carbon like that. It may be condensed to form the fatty acids. Okay. Fatty acid is an anabolic process. Ultimately, it need an energy source like ATP and NADPH will be utilized. Okay. Apart from that, enzymes are required for fatty acid synthase. Okay. The primary enzyme involved in fatty acid synthase is fatty acid synthase complex. Okay. Fatty acid synthase complex. This is another important and repeated objective question. Okay. Now we are going to see about the enzyme. Okay. Fatty acid synthase, what are the different components in the fatty acid synthase, everything we are going to discuss now. Okay. As I told the acetyl CoA will act as a precursor for fatty acid synthesis. Okay. If you remember, in TCA cycle, okay, in TCA cycle, acetyl CoA will be there. Okay. Then if you see properly, after the glycolysis, pyruvate will form. This pyruvate will be converted into acetyl CoA. That will be happen in mitochondrial matrix. So the fatty acid biosynthesis is happening in cytoplasm or plastid. So how this acetyl CoA will be transported to the cytoplasm? This is one of the repeated question. How the acetyl CoA is transported from mitochondrial matrix to cytoplasm. Okay. There is a shuttle known as citrate shuttle. Okay. Shuttle will happen. This shuttle will helps in taking the acetyl CoA molecule from mitochondrial matrix to cytoplasm. This is very much important question. From this citrate cycle, what question they will ask means, what is the number of ATP molecule matrix to cytoplasm? Okay. Total of 2 ATPs will be utilized. 2 ATP will be utilized. Okay. If you see here, acetyl CoA is present in mitochondria. This will be converted, this will join with oxaloacetate to form citrate. The citrate will come to the cytoplasm by the action of citrate lyase. This is another important thing. By the action of citrate lyase, acetyl CoA molecules will be released to the cytoplasm. This may be utilized for fatty acid synthesis. Okay. Then if you see the further step, one more ATP will be utilized. One more ATP will be utilized by pyruvate carboxylase. So, at, in an average, if you see, 2 molecules of ATP will be utilized in citrate cycle for transferring a molecule of acetyl CoA from mitochondria to cytosol. This is most repeated question you have to remember. Okay, 2 molecules of ATP will be utilized. Okay. Now, the acetyl CoA molecules will be present in the cytoplasm. Okay. After the citrate settled. Okay. Now, how this acetyl CoA molecules are condensed to form fatty acid? Okay. Here comes the role of enzyme fatty acid synthase. This is very important enzyme. Okay. This fatty acid synthase complex, why we are saying complex means 7 different enzyme complex will be present in that. That means 7 different domains will be present in a single polypeptide. Okay. 1, 2, 3, 4, 5, 6, 7. Total 7 different domains will be there in single polypeptide enzyme. Okay. These are the different domains. If you see, ketoacyl ACP synthase, malonyl acetyl CoA ACP transferase, hydroxyacyl CoA dehydratase, enoyl ACP dehydratase. Okay. ACP, then ketoacyl ACP reductase. You have to just remember what are the different domains. This is also another important thing. Okay. Total 7 different domains will be there. This is another important point you have to remember. Okay. Now we are going to see the step by step how the fatty acid will be synthesized from acetyl CoA. Okay. The committed step for synthesizing of fatty acid is what means. First, the acetyl CoA molecule will be binded with carbon dioxide to form malonyl CoA. Okay. That will be catalyzed by acetyl CoA carboxylase enzyme. Okay. This is the first commitment step. In first commitment step, acetyl CoA will be converted into malonyl CoA by the action of acetyl CoA carboxylase. This is a carboxylase enzyme. So, biotin will act as an cofactor. Okay. These two are key important points. Okay. These two are key important point. Acetyl CoA carboxylase will catalyze the first commitment step for fatty acid synthesis. Okay. Second thing, it is a biotin dependent enzyme. Okay. So, acetyl CoA will be converted into malonyl CoA. Okay. This is a 3-carbon compound. This is the first commitment step. Okay. If you see the upcoming step, okay, there is total 4 steps are there. This will be repeated for extending the 2-carbon compounds. Okay. For example, a 2-carbon acetyl CoA will be there. After this 4 reaction, this will be converted into 4-carbon compounds. Okay. Further, next 4 step will happen. That will convert to 6-carbon compound. Like that, the cycle will be repeated to form the fatty acid. For example, 12-carbon fatty acid, 14-carbon, 16 carbon, 18 carbon, everything will be produced by repeating the same cycle. Okay, repeating the same cycle. This is the key point you have to remember. Total 4 reactions are there. What are that 4 reactions? First is condensation, second is reduction, third dehydration, and fourth is reduction. Okay, these are the 4 steps. They have asked one time this sequence. Okay, what is the sequence that are involved in fatty acid synthesis? Okay, you just observe here. First acetyl ACP will be there. That will bind with malonyl ACP. Okay, that is condensation reaction. Here one CO2 molecule will be released. So total 4 carbon compound will be formed here. That is acetoacetyl ACP. Okay, as I told, the fatty acid synthesis is an anabolic process. So the reducing equivalent NADPH will be utilized there. That will lead to formation of D-beta hydroxybutyl CoA. Okay. After that, other enzyme will be further act on this. That will be lead to the formation of 4 carbon fatty acid, that is butyric acid. Okay. I hope this is clear. These are the key points you have to remember. Okay. Total 4 steps will be there: condensation, reduction, dehydration, and again reduction. So in each cycle, 2 molecules of NADPH will be utilized. This is also another important point you have to remember. Okay, 2 molecules of NADPH will be utilized. I hope this is clear for everyone. Okay, I know so much enzymes are there in metabolism. You have to remember the key important points only. Okay, for example, acetyl CoA carboxylase is the committed enzyme for fatty acid synthesis. Like that, key important point only you have to remember. Okay. So now 4 carbon butyric acid was formed. Then again one more cycle, the same cycle will be happened. Okay. Then that 4 carbon fatty acid will be converted into 6 carbon. If one more cycle will happen, means that 6 carbon will be converted into 8 carbon. Like this, the fatty acid synthesis will be happen. I hope this is clear for everyone. Okay. So fatty acid synthesis will be happen like this only. Okay. Now how the double bonds will be introduced in the fatty acid. For example, if you see 18 carbon fatty acid is what? Oleic acid will be there. This is 18 carbon. If you want to introduce a double bond, that is 18:1. Okay. This is what? Sorry, 18 carbon is stearic acid. Stearic acid. Okay, 18:1 is oleic acid. Okay. So if you want to introduce this double bond, desaturase enzymes are required. Okay, desaturase enzymes are required. That will be particularly present in smooth endoplasmic reticulum. This is another important point. Okay, desaturase is the enzyme that will be involved in formation of double bonds. Okay, desaturase is the enzyme that will be involved in formation of double bond. This enzyme will be present in smooth endoplasmic reticulum. Okay, these are the key points you have to remember. Now we have completed the fatty acid synthesis. How the fatty acid synthesis is happening. Okay, how the double bond will be introduced. Okay, now we are going to see how the regulation will happen. Okay, as I already told, acetyl CoA carboxylase is the key enzyme. Okay, so if this acetyl CoA carboxylase is present in phosphorylated form, that will become inactive. This is the point you have to remember. Okay. Acetyl CoA carboxylase, if it is present in phosphorylated form, that will become inactive. This is the key regulation step. Okay. Okay. I hope up to this everything is clear for all the persons. Any doubt is there? Fatty acid synthesis. Any doubt? Kindly let me know. Any doubt in fatty acid synthesis? Hmm? You guys are listening now? Any doubt? No, sir. Yes. Yes, yes, yes. So we have completed now fatty acid synthesis. Okay. Now we are going to see how these fatty acid molecules are assembled to form triacyl glycerol. Okay. We know first we will see what is meant by triacyl glycerol. Triacyl glycerol means what? Glycerol backbone will be there that will be bind with 3 molecules of fatty acid. Okay. So from where this glycerol will come? This fatty acid was already synthesized. Okay. This we have to know. This glycerol, okay, glycerol will come from glycolysis product. If you see dihydroxyacetone phosphate, will be there from glycolysis. This will be converted to glycerol 3 phosphate by the enzyme dehydrogenase. Okay? By the action of dehydrogenase enzyme, this dihydroxyacetone phosphate will be converted to glycerol 3 phosphate. Okay? If this glycerol 3 phosphate is binded with one molecule of fatty acid, that we will call as lysophosphatic acid. Okay? Lysophosphatic acid. You just remember the structure here. First carbon, second carbon, and third carbon of glycerol. Here in first carbon, fatty acid is attached. Then third carbon, phosphate group is there. If one more fatty acid was attached with this lysophosphatic acid, that we will known as phosphatic acid. Okay. So glycerol backbone is there. Two fatty acid is binded here. Then one phosphate group is there. If phosphatase enzyme will act on this, the phosphate group will be removed. That will lead to formation of diacyl glycerol. Okay. If one more fatty acid will come and bind here, triacyl glycerol will form. Okay. Here the question we can expect, what is the question they will ask means, what is mean by lysophosphatic acid? Okay. What is mean by lysophosphatic acid? This you have to know the structures. What are the structures? 3 carbon will be there of glycerol. Okay. At first carbon, fatty acid will be there. Third carbon will have phosphate group. That is known as lysophosphatic acid. Okay. Now we are going to see how the membrane lipids are biosynthesis. Okay. This is most repeated question. Okay. Most repeated question and basic also. Okay. What are the classifications of phospholipid? From here, sure you will get one question. Okay. If you see the classification of phospholipid, based on the head group, the phospholipids are classified into phosphatidylethanol choline, serine, phosphatidylglycerol, phosphatidylinositol, and cardiolipin. This is the most repeated question. Okay. Cardiolipin will be highly present, that is most abundantly present in membranes of mitochondria. Okay. Mitochondrial membrane will have this cardiolipin. Okay. This is most important point. And another important thing what you have to remember is, what is the head group of cardiolipin? The head group attached in the cardiolipin is phosphatidylglycerol. Phosphatidylglycerol. This is another important point you have to remember. Okay. Now we are going to see how the phospholipids are synthesized. Okay. We already seen phosphatic acid means glycerol backbone will be there. Fatty acids will be attached with first carbon and second carbon. Third carbon will have only phosphate group. Okay. Here if head group is attached, this is known as phospholipids. Based on this head group, they have classified different types. Okay. Now, if you see the membrane lipid biosynthesis, there are two different pathways are there. Okay. Diacylglycerol activation and head group activation. You just have to remember the lipids, that is phosphatidylcholine and phosphatidylethanolamine, will be synthesized from both the pathway. Remaining lipids will be synthesized only by the strategy 1. These are the things you have to remember. Okay. Phosphatidylcholine and ethanolamine will be synthesized from both the pathway, but remaining things will be synthesized only from strategy 1. This is the important thing you have to remember. Okay. Here just I have given how the lipids are, how the different membrane lipids are synthesized. You just have to go through once. Okay. What are the different head groups? Okay. This is not that much important. You just go through once. Okay. Next most important thing is sphingolipids. Okay. Sphingolipids. We have completed now phospholipids. Now we are going to see about sphingolipids. Okay. Sphingolipids, the backbone of sphingolipids are ceramide. Okay. Ceramide is the backbone of sphingolipid. If you see phospholipids, okay, phosphatic acid will act as a backbone. Okay. Then the another most important thing, if you see here, the fatty acid will linked in the phosphatic acid through ester linkage. Okay. Ester linkage. Okay. But the fatty acid in sphingolipid will be linked by amide linkage. This is the key difference between sphingolipids and phospholipids. This question we can expect. Okay. There are different classifications are there. Sphingomyelins, okay. Then cerebrocytes, globocytes, and gangliocytes. You just remember sphingomyelins, cerebrocytes, globocytes, and gangliocytes. Globocytes and gangliocytes. Okay. You have two gangliocytes. Okay. You have to remember what is mean by cerebrocyte, what is mean by globocyte, and what is mean by gangliocyte. From here, yeah, from here, sure you will get the question. Okay. From here, sure you will get the question. Okay. So cerebrocyte means the head group will be present as sugars. Okay. Sugars will be there. Globocytes means sugar will be there, but more than one sugar will be there. Okay. If only one sugar is there means that is cerebrocyte. If more than one sugar is there means that is globocytes. Then gangliocytes means there will be charged sugars will be there. Particularly if you see negative charged sugars will be there. Okay. For example, sialic acid will be there in gangliocytes. These are negative charged molecule. I hope this is clear. And the most important thing, phosphorus containing sphingolipids. Okay, the only sphingolipids having phosphorus in its structure is sphingomyelin. Okay, this is JRF 2021 question. Okay, as well as they have asked in SRF question also this one. Okay, so sphingomyelin is the only sphingolipid having phosphorus in its backbone. Okay, this is another important thing you have to remember. Now we are going to see how the sphingolipids will be synthesized. In earlier slide, we have seen what are the classification. Now we are going to see the sphingolipid synthesis. Here, the only one thing you have to remember is the sphingolipids was synthesized from the backbone of the sphingolipids will be synthesized from palmitoyl-CoA and serine molecule. This is enough to read about the biosynthesis of sphingolipids. Okay. So palmitoyl-CoA and serine will act as a precursor for synthesis of sphingolipids. Okay. This is the most important thing you have to remember. Okay. Now we have completed what is meant by how the fatty acids are synthesized. Okay. How fatty acids are synthesized, how triacylglyceryls are synthesized, what are phospholipids and what are sphingolipids and how they are synthesized. Okay. Now we are coming to last part of synthesis. Okay. Signaling molecules, particularly if you see eicosanoids, the most repeated question, eicosanoids. Okay. The precursor for eicosanoid synthesis is arachidonic acid. This is very important. Arachidonic acid is the precursor for synthesis of eicosanoids. Okay. If you see here, there are three classification of eicosanoids are there. These are the things you have to remember. Three classifications are there. First is prostaglandins. Prostaglandins. Okay. Second is thromboxanes. Thromboxanes. And third is leukotrienes. Leukotrienes. These are the different classes of signaling molecules that are eicosanoids. Okay. Among these three, prostaglandin and thromboxane are cyclic structured molecule. They are cyclic structured molecule. Okay. Leukotriene L, that is linear molecule. This is the thing you have to remember. Okay. Thromboxanes and prostaglandins are cyclic molecule. Then leukotriene is a linear molecule. This is the thing you have to remember. Yes, voice is not clear. Video is lagging. Network issue or what? Video is lagging like that. Network issue or what? Or some problem is going on. Actually, I'm getting this message from so many times. Whether I am audible now? Whether my voice is clear? Kindly let me know. Hello students, whether my voice, yeah, voice is clear. Yes. Yeah, voice is clear now. Yes. Sorry for the disturbance. Okay. Again, we are going to see eicosanoids. Yeah, eicosanoids. Okay. This is a class of signaling molecule. Okay. It's a signaling molecule. We already discussed the precursor for eicosanoids is arachidonic acid. Okay. There are three different eicosanoid classifications are there. Prostaglandin, thromboxane, and leukotriene. Okay. Among this, prostaglandin and thromboxane are cyclic molecule. Leukotriene is linear molecule. This is the thing you have to remember from eicosanoids. Okay. With this, we are completing our synthesis part. Okay. How the fatty acid metabolism synthesis is happening. Okay. Fatty acid synthesis, triacylglycerol synthesis, phospholipids, sphingolipid, and signaling molecule biosynthesis we have completed. Okay. Now I am going to ask one question. Which is the sphingolipid that is having phosphorus in its structure? Kindly respond your answer. What is the sphingolipid that is having phosphorus in its structure? Option A, cardiolipin. Option B, sphingomyelin. Option C, cerebrocytes. Option C, cerebrocytes. Option D, gangliosides. What will be the answer? What is the sphingolipid that is having phosphorus in its structure? Yes, very good. Excellent. Excellent. Sphingomyelin. Excellent. Okay. Yes, now we are going to start fatty acid catabolism. Yes, now we are going to start fatty acid catabolism. Okay, how the fatty acids are catabolized to produce energy in our body. Okay, how the fatty acids are catabolized to produce energy in our body. Okay, the major pathways. Okay, so there are different pathways are there for fatty acid catabolism. Okay, alpha oxidation, beta oxidation, omega oxidation, like that different pathways are there. Okay. The major pathway for beta oxidation is pathway for fatty acid oxidation is beta oxidation. This you have to remember properly. Okay. So beta oxidation is the major pathway for fatty acid oxidation in mammals. This is another important thing. Okay. In mammals, beta oxidation will be major pathway. Okay. Okay, this is clear. Then another most important thing, this beta oxidation will be primarily happen in mitochondria. Okay, this will be happen in mitochondria of mammals, but in plants it will happen in peroxisome. These two are most important and repeated question they are asking. Okay, so beta oxidation will be happen in mitochondrial of mammals, peroxisome of plants. Okay. Now we are going to see what is mean by beta oxidation. Why we are saying beta oxidation? Okay, everything we are going to see. Okay, this beta oxidation is the major pathway for fatty acid catabolism. Okay, if you see the fatty acids are there. For example, 6-carbon fatty acid are there. Okay, just I am writing here. This is a 6-carbon fatty acid. In every cycle of beta oxidation, 2 carbon molecules will be removed from the existing fatty acid in the form of acetyl CoA. If this concept is clear, the fatty acid catabolism, entire fatty acid catabolism will be, yes, video again it's having problem. TK, guys, you take 5 minutes rest. TK, we will resume at 5:35. TK, I will rectify this problem, then I will come. will try. Whether I'm audible, students? Whether I'm audible? Yes, yes, very good. If again network issue is there means kindly let me know. Okay? Yes. Yeah. Now we are going to see about fatty acid catabolism. Fatty acid catabolism. Okay? Yes. Yes. Okay? Now fatty acid catabolism. Again we are going to restart. Okay? Fatty acid catabolism. Guru, Guru. I hope now network issue is not there. Kindly let me know whether any lagging is there in video. Whether I'm audible. Kindly let me know whether I'm audible. Yes, students, kindly let me know whether I'm audible. No, 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 no. Again, it's disconnecting. After 1 or 2 seconds, again it's disconnecting. Okay. Whether I'm audible now, students? Kindly let me know whether I'm audible. Yeah. Now we are going to see about fatty acid catabolism. Okay, still now we have seen how the fatty acids are synthesized and how it is converted to lipids. Okay, for example, how it is converted to sphingolipids and phospholipids, everything we have seen. Now we are going to see about fatty acid catabolism. Okay, how the fatty acids are catabolized in plants as well as in mammals to produce the energy. Okay, as the name itself is there, it's a catabolic process primarily utilized for the synthesis of ATP. Okay, primarily utilized for the synthesis of ATP. Okay, if you see the major pathway of fatty acid catabolism, beta oxidation is the major pathway for fatty acid catabolism. Why we are saying this as a beta oxidation? That also we have to know why we are seeing that as beta oxidation. If you see the fatty acid, for example, I am writing the structure of 4 carbon fatty acid. Okay. This is 4 carbon fatty acid. Okay. COH will be there. This is 4 carbon fatty acid. If you see in each cycle of beta oxidation, 2 carbon will be removed in the form of acetyl CoA. Why we are saying this as beta oxidation means the beta carbon will be cleaved. Okay, the beta carbon, the bond between the beta carbons will be cleaved. For example, this they have asked one time. The bond between second and third carbon will be cleaved in beta oxidation. That is, beta carbon will be cleaved. Then the most important question what they will ask is, what is the molecules that will be released in every cycle of beta oxidation? Okay. So in every cycle, a molecule of acetyl CoA will be released. Okay. A molecule of acetyl CoA will be released. This will be entered to TCA cycle to produce NADH and FADH. NADH and FADH2 molecules along with ATP will be produced. Okay. Okay. Now we are going to see how the fatty acids are converting to acetyl CoA. Like fatty acid synthesis, I told in fatty acid synthesis, acetyl CoA carboxylase is the commitment reaction. Okay. Like in fatty acid oxidation, the commitment reaction will be catalyzed by fatty acyl CoA synthetase enzyme. CoA synthetase enzyme. Okay. Again, the name itself it's there. It's a synthetase enzyme. That means it requires ATP. Okay. So why this fatty acyl CoA synthetase enzyme is coming here? You have to remember. Okay. The free fatty acid, whatever the free fatty acid is there, that will be bind with the CoA molecule to form fatty acyl CoA pools. Okay. Fatty acyl CoA pools will be created. This fatty acyl CoA pool will be entered into beta oxidation. I hope this is clear. The most important thing is fatty acyl CoA synthetase is the first commitment reaction. Okay. As I already told, mitochondria is the primary site for beta oxidation. So, whatever the fatty acid that are present in cytoplasm has to be transferred to mitochondria for beta oxidation. There comes the role of carnitine shuttle. Okay. Carnitine shuttle. This is also another repeated question. In carnitine shuttle, what it will happen means the fatty acyl pool, for example, fatty acyl CoA will bind with the carnitine to form acyl carnitine will be formed. Okay, acyl carnitine. Acyl means what? It's a fatty acid. Fatty acid with carnitine molecule will form. This will be shuttled to the mitochondrial matrix. Okay. Here the question what they will ask means the carnitine acyl transferase enzyme 1 and carnitine acyl transferase enzyme 2. These are the primary enzymes involved, primary transporters involved in transporting the acyl cool acyl CoA pool from cytoplasm to mitochondrial matrix. Okay. And another most important thing what they will ask means this carnitine shuttle will be required only for transporting medium chain and long chain fatty acids. Okay. Short chain fatty acid can directly enter into the mitochondrial matrix. These are the key points you have to remember. Okay, these are the key points you have to remember. Now we are going to see the steps of beta oxidation. Okay, what are the 4 steps of beta oxidation? This is JRF 2023 question. They have asked the sequence of reaction that will happen in beta oxidation. See, sequence of reaction happen in beta oxidation. As I already told, in every cycle of beta oxidation, acetyl CoA molecule will be released. That acetyl CoA will enter to TCA cycle. Because of this enormous amount of FADH and NADH molecule will be produced. That will go to electron transport chain for production of ATP. Okay. These key steps are very important. You just remember. Okay. The steps in fatty acid oxidation, that means particularly beta oxidation is very important question. Okay. There are 4 steps are there. Okay. Step 1, 2, 3 and 4. 4 steps are there. You just observe here in first step, fatty acyl CoA dehydrogenase enzyme will come. Okay. First acyl CoA dehydrogenase enzyme will come. Okay, dehydrogenase enzyme will come. In first step, one molecule of FADH will be produced. Okay, one molecule of FADH will be produced. Okay, then the product formed by this enzyme is trans del 2 enyl CoA. Trans del 2 enyl CoA. This trans del 2 enyl CoA, here water molecules will be added to form L beta hydroxy acyl CoA. This is another important question. L beta hydroxy acyl CoA will be formed after the completion of second step. In third step, if you see, one molecule of NADH will be produced by the action of enzyme dehydrogenase. So, if you see in this fourth step, one molecule of NADH and one molecule of FADH will be produced per each cycle. This is another important point you have to remember. One molecule of NADH and one molecule of FADH will be produced per cycle of beta oxidation along with a molecule of acetyl CoA. This is the key point they will ask. Okay. So, one molecule of NADH and FADH will be produced per cycle along with an acetyl molecule. Okay. There are 4 steps in beta oxidation. Okay. Now we are going to calculate. What is the amount of energy will be produced if a fatty acid molecule is catabolized through beta oxidation. This is most repeated question. Okay. If you see, for example, I have 14 carbon fatty acid. Okay. So 14 carbon means okay, divided by 2. So 7 will come. So at the at the 6th cycle, 2 molecule of acetyl CoA will be released. Okay, 2 molecule of acetyl CoA will be released. So if a 14 carbon fatty acid is there, means 6 cycle of beta oxidation will happen, because at the 6th cycle 2 molecule of acetyl CoA will be released from this 4 carbon compound. This is very important thing. Okay, they can give, if a 20 carbon fatty acid is entering beta oxidation, how much number of beta oxidation cycle has to be happen. The formula is n by 2 minus 1. That means n is what? N is number of carbon atom. So 10 minus 1. So 9 cycle is needed for 20 carbon fatty acid. This is another important thing you have to remember. Okay. Now we are going to do the energy calculation. Okay. We know acyl CoA dehydrogenase enzyme will produce FADH molecule. Beta hydroxy acyl CoA dehydrogenase will produce FADH molecule. So each cycle will produce one NADH and one FADH2. That means if palmitic acid is 16 carbon fatty acid, this will go 7 number of beta oxidation cycles. So 7 molecule of FADH, 7 molecule of NADH will be produced directly from beta oxidation. Okay. Apart from that, 8 molecule of acetyl CoA will be there. If that will enter to TCA cycle, this will produce an enormous amount of energy. This calculation is very important. On an average, if you see, the net gain will be around 108 ATPs. Okay. So 108 ATP will be produced if the palmitic acid is entering to beta oxidation. This is most repeated question. 108 ATP will be produced by beta oxidation of palmitic acid. Okay. This is total ATP produced. Okay. This is very, very important thing. Okay. Now we are going to see other minor pathways. Okay. Now we are going to see the fatty acid oxidation, that is beta oxidation of unsaturated fatty acid. So far we have seen about unsaturated fatty acid. Now we are going to see about monounsaturated fatty acid and polyunsaturated fatty acid. What is this unsaturated fatty acid? That you have to remember. Okay. Unsaturated fatty acid means that will have presence of double bond. The double bond in fatty acid will be always in cis position. Okay. But the product, if you see in beta oxidation, the fatty acid will be present in trans position. So the cis to trans conversion should be there. Okay. Here comes the role of trans del 2. This is important enzyme. Del 3 to del 2 enyl CoA isomerase. This is very, 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 very important question. This they have asked continuously. JRF 2021, 2022 and 2023. They have asked one time in statement question, one time in match the following question. Okay, so this is very, very important thing. Okay, for oxidation of monounsaturated fatty acid, we need del 3 del 2 enyl CoA isomerase enzyme. Del 3 del 2 enyl CoA isomerase enzyme. For oxidation of polyunsaturated fatty acid, we need two enzymes. Okay, one is del 3 del 2 enyl CoA isomerase. Okay, del 3 del 2 enyl CoA isomerase. Along with we need 2,4-dienyl CoA reductase. 2,4-dienyl CoA reductase. These are the two enzymes that are additional enzymes required for oxidation of polyunsaturated fatty acid. Okay, I hope this is clear. Now again important thing. Okay, again important thing. How the oxidation of odd carbon fatty acid will happen? How the oxidation of odd carbon fatty acid will happen? If you see this propenyl CoA is a 3-carbon fatty acid. How this propenyl CoA is oxidized to other products? This is another important question. Okay, propenyl CoA by the action of carboxylase, it will convert into dimethylmalonyl CoA. Okay, this is 4-carbon compound. Okay, one time they asked what is the cofactor for propenyl CoA carboxylase. Okay, so that biotin will be the cofactor for propenyl CoA carboxylase. Okay, this dimethylmalonyl CoA will be converted into L-methylmalonyl CoA. This will be rearranged to form succinyl CoA. The succinyl CoA will enter to Krebs cycle. Okay. Here what they will ask means what is the cofactor for methylmalonyl CoA mutase. This is also another important thing. The cofactor for methylmalonyl CoA mutase is coenzyme B12. That is vitamin B12 will act as a cofactor. This is very, very important question. Okay. You shouldn't forget. So if beta oxidation of hard carbon fatty acid is happening means end product forming is succinyl CoA, then cofactor for propenyl CoA carboxylase is biotin, and cofactor for coen and methylmalonyl CoA mutase is coenzyme 12. Okay, I hope this is clear. I know so much of enzymes and cofactors are there in biochemistry, but these all are very important questions. Okay, whatever is important that I am saying, you have to remember that properly. Okay, read properly and revise properly. This is most important thing. Okay. Now, this is coordinate regulation of fatty acid synthesis. This we have already discussed. By the phosphorylation of acetyl CoA carboxylase, the regulation will be happen. This we already discussed. Okay. Now we are going to see peroxisomal beta oxidation. That means particularly in plants, the major site of beta oxidation is peroxisome. Okay. What are the key difference between peroxisomal and mitochondrial beta oxidation? That we are going to see. Okay. In peroxisomal beta oxidation, in first step, FADH will not be produced. Directly hydrogen peroxide molecule will be produced. Okay, this is first point. First point is FADH2 will not be produced. Second point, if you see, acetyl CoA formed in the peroxisome will be transported to the mitochondria for beta oxidation. Acetyl CoA will be transported to mitochondria for beta oxidation. These two are very, very important point. Okay, this two is enough. And third most important thing, the substrate for beta oxidation in peroxisome is long chain fatty acids. Okay, long chain fatty acid. They are very long chain fatty acid will act as a substrate. Here, medium chain fatty acid will act as a substrate in mitochondria. These are the key important difference you have to remember. Okay. Apart from the two minor pathways are there, that is omega oxidation and alpha oxidation. Okay. I am going to say the exam point of questions only. Okay. In omega oxidation, what it will happen means, in spite of this alpha, in omega oxidation will happen from the omega carbon. Okay, omega carbon oxidation will happen. Okay. The question what they will ask means omega oxidation will be primarily happen in endoplasmic reticulum, and the product formed will be dicarboxylic acid will be formed. Product will be dicarboxylic acid. Product is dicarboxylic acid. Okay. dicarboxylic acid. These are the key point you have to remember. And last pathway is alpha oxidation. Last pathway is alpha oxidation. If you see in alpha oxidation, the beta carbon will have some of the additional group, methyl group is there. If the beta carbon is occupied with some of the group, the alpha oxidation will happen. Okay. You have to remember only one thing in alpha oxidation. Okay. If the alpha oxidation enzymes, particularly if you see phytanyl-CoA hydroxylase enzyme is deficient, that will lead to formation of refsum disease. Okay. Refsum disease. This is very important question. So refsum disease will be caused due to defect in the enzymes of alpha oxidation. Okay. Refsum disease. I hope this is clear for everyone. Okay. Any doubts in fatty acid catabolism? Any doubts? Kindly let me know. Any doubts is there? Any doubts? Safura, any doubt? Then Kavian, any doubts? Priya, okay. If no other doubts means I will start. Okay. Now we are going to see about ketone body synthesis. Okay. How the ketone bodies will be synthesized. Okay. Ketone body synthesis. Here the most important thing what you have to know means what are the examples of ketone bodies. Okay. Example of ketone bodies. Three things are there. Acetone, acetoacetate and beta hydroxybutyrate. Acetone, acetoacetate and beta hydroxybutyrate. These are the ketone bodies. The ketone body synthesis will be primarily happened in mitochondria of liver. This is another important thing. Mitochondria of liver. These pathways are not important. Just you remember acetone, acetoacetate and beta hydroxybutyrate are major ketone bodies. Okay. This ketone body synthesis will primarily happen in mitochondria of liver. If you see the ketone body utilization, the utilization will be always happen in extra hepatic tissues. That means liver is only the producer of ketone bodies, but liver cannot utilize that ketone bodies. Why means due to the lack of enzyme thiophorase. This is also another important point you have to remember. Due to lack of the enzyme thiophorase. Okay. With this, we have completed the lipid metabolism. Now we are going to start amino acid biosynthesis and amino acid catabolism. This is only minor topic. We are going to discuss the important points only. Okay. If you see the structure of amino acid, that's amino acid will have, just I am writing here the structure. Central carbon will be there. This will have hydroxy group, sorry, carboxy group, amino group will be there. Along with that R group and H will be there. Okay. So nitrogenous component also will be there in amino acids. Okay. Now we are going to see how the amino acid biosynthesis will be happened in plants as plants. Okay. How the amino acid biosynthesis will happen. Okay. In plants, all the 20 amino acid will be synthesized. Okay. But in humans, only non-essential amino acid synthesis will be happen. Essential amino acid synthesis will not happen. Okay. If you see the major carbon sources, the carbon sources are there now. These carbon sources are provided by byproducts of glycolysis, TCA cycle and pentose phosphate pathway. Sorry, intermediates, not byproducts. Intermediates of glycolysis, TCA cycle and pentose phosphate pathway will provide the carbon skeleton for fatty acid, sorry, carbon skeleton for amino acid biosynthesis. Okay. Now we are going to see what are the carbon skeletons, what are the carbon skeletons or what are the intermediates from where the amino acids are synthesized. Okay. Before going that, this is another important table you have to remember. What are the essential amino acid, non-essential amino acid and conditionally essential amino acid. Okay. You have to remember this 9 essential amino acids. Okay. Histidine, isoleucine, leucine, methionine, lysine, phenylalanine. See, just you remember aromatic amino acid, phenylalanine, tyrosine, tryptophan. ठीक है. Phenylalanine, threonine, tryptophan. Apart from that, valine, leucine, isoleucine, histidine, and methionine. These are the essential amino acid. You just remember this one. Okay. From here, they can ask what is the essential amino acid. Like that, they may ask. Okay. If you know the essential amino acid, we can easily know what are the non-essential amino acids. Okay. If you see the pathways, okay, there are total 6 families are there. Why we are seeing 6 families means six intermediates, 6 different intermediates from TCA cycle, glycolysis and pentose phosphate pathway will act as a precursor for synthesis of amino acids. Okay. These are the 6 families you just remember. From alpha ketoglutarate, four amino acid will be synthesis: glutamate, glutamine, proline, and arginine. Okay. This is important. Okay. This is the point. This is the thing they are going to ask the question. If they want to make question paper difficult, they will ask question from this parts only. Because most of the people will omit amino acid metabolism and nucleotide metabolism. Okay. These both are important, but okay. Then 3-phosphoglycerate, okay, GSC. You just remember GSC. Glycine, serine, and cysteine will be synthesized from 3-phosphoglycerate. Okay. Oxaloacetate will act as a precursor for aspartate, aspargine, methionine, threonine, and lysine. This is very, very important. Okay. What are the amino acids that are synthesized from oxaloacetate? And if you see aromatic amino acid, phenylalanine, tyrosine, and tryptophan will be synthesized from phosphoenol pyruvate and erythrospore phosphate. Okay, phosphoenol pyruvate and erythrospore phosphate. Ribose 5 phosphate from ribose 5 phosphate from pentose phosphate pathway will act as a precursor for histidine. I hope this is also clear. Then pyruvate that will act as a precursor for alanine, valine, leucine, and isoleucine. Okay. These are the 6 major biosynthetic family for amino acid. Here we can ask match the following question. They can ask match the following question. Which amino acid is synthesis from which precursors? This is very important point you have to remember. Okay. Now we are going to see. We have seen what are the sources for carbon. Now we are going to see from where the amino group in the amino acid will come. Okay. There are 2 different pathways are there from where the amino source will come. That is N source. First thing is transamination reaction. This is repeated question. The cofactor for transamination reaction is PLP, pyridoxal phosphate will act as the cofactor. This is most repeated question. Okay. Transamination cofactor is PLP. In transamination, what it will happen means alpha keto acids will be there. The amino group from one amino acid will be donated to the alpha keto acid. Because of this, new amino acid will be formed. I hope this is clear. In transamination, one of the amino acid is donating the amino group to carbon compound that will lead to formation of amino acids. That is new amino acid will be formed. Okay. Then second reaction, then second way by which the ammonia group will be donated is reductive amination process. Reductive amination process. If you see here, alpha keto acid will bind with ammonia. Okay. The source of the energy for reductive deamination is NADPH and the enzyme involved is glutamate dehydrogenase. This is another important point, GDH. Okay, by the action of GDH, amino acids will be formed. I hope this is clear for everyone. So, 2 pathways are there. Okay, how the amino group will be produced. Okay, one is reductive amination and second is transamination reaction. Then here I just have listed what are the biomolecules or what are the molecules that are derived from amino acid. Like for example, you have to just remember from tyrosine, dopamine, norepinephrine and epinephrine will be produced. No need of remembering the pathway. You just have to remember from tyrosine, dopamine, norepinephrine and epinephrine will be produced. And from glutamate, GABA will be produced. Gamma amino butyric acid will be produced. Okay. Histidine will act as a source for histamine production. From tryptophan, serotonin will be produced. These all are very, very important point. Okay. The molecules derived from amino acid. From here we can expect one question. Okay. And another most important thing, tryptophan It will also act as precursor for indole 3 acetic acid. So plant hormone synthesis will be there. Phenylalanine for cinnamate. Okay. Then this is JRF 2019 question. JRF 2019. Succinyl-CoA. Succinyl-CoA will act as the precursor for heme. Okay. Act as a precursor for heme. Succinyl-CoA plus glycine. Okay. Glutamate in plants, glutamate tRNA will act as the precursor for heme biosynthesis. I hope this is clear. Succinyl-CoA plus glycine will be involved in synthesis of heme in mammals. Glutamate or glutamal tRNA will be act as a precursor for heme biosynthesis in plants. Okay. Like this, I have just listed the molecules. You just see once. Okay. From here, we can expect question. Okay. What are the biomolecules produced? Okay. Then from amino acid. Okay. From amino acid, how the purines and pyrimidines nucleotides are synthesized. This also I have listed here. This we are going to see briefly in the upcoming slides. Okay. From where which amino acids will act as a carbon and nitrogen source for each and every nucleotides we are going to see in our nucleotide metabolism topic. Just that's why I am just skipping here. Okay. Now we are going to see how the amino acids are catabolized. If amino acids are catabolized, what it will happen? Two things will happen. The amino group will be released separately. This will be released. This will be released through urea cycle from the body. Okay. And whatever the carbon sources is there, this can be utilized for energy production. Okay. This is the amino acid catabolism pathway. Okay. Based on how the amino group is excreted by an organism, they can classify the organism into different sources. Okay. One is aminotellic organism. That means it will excrete the ammonia directly. That is aminotellic organism. Second is ureotellic organism. What is ureotellic? Means the ammonia will be converted into urea. This is non-toxic form and it will be excreted from the body. Okay. And third most important thing is uricotellic organism. That means uric acid will be excreted. Uric acid will be excreted. The example of uricotellic organism example is what? Birds and reptiles. Birds and reptiles. Okay. Now we are going to see the urea cycle. From urea cycle, they have asked one question in JRF 2023. Okay. The major organelles involved in urea cycle is what? Cytoplasm and mitochondrial matrix. These are the organelles involved in urea cycle. I am not going to explain the each and every pathway. Just I want to say the important things. Okay. In urea cycle, 4 molecules of ATP will be utilized. This is the key important point you have to remember. 4 molecules of ATP will be utilized. The organelle involved in cytoplasm and mitochondria. These 2 points are highly sufficient for JRF examination. Apart from that, if they want to ask more deep question means the regulatory enzyme in urea cycle is carbamyl phosphate synthase 1. Okay, carbamyl phosphate synthase 1. I am just writing CPS1. CPS1 is the major regulatory enzymes of urea cycle. Now we are going to see how the urea cycle is linked with citric acid cycle, because if you see in urea cycle, during urea cycle, fumarate will be released. How this fumarate is entering to TCA cycle they have given here. This is just for your understanding only. Just go through once. Okay. This is the summary of amino acid catabolism. For example, different amino acids they have given. How it is entering to intermediates of the pathways they have given here. The most important thing what you have to remember means what are the glucogenic and ketogenic amino acids. These things you have to remember. What are the examples of glucogenic and ketogenic amino acid? That means the carbon atom that are involved in the formation of glucose molecules, that are glucogenic amino acids. Okay. You have to remember this. This is the key important things you have to learn from amino acid catabolism. Okay. 5 amino acid will act as both glucogenic and ketogenic. This is very important. Okay. Threonine isoleucine, phenylalanine tryptophan and tyrosine. This is very important. PITT. Okay. phenylalanine, isoleucine, tyrosine, tryptophan and threonine. Okay. Leucine and lysine are solely ketogenic. Leucine and lysine are solely ketogenic. These two things are very, very important. You have to remember. I hope this is clear for everyone. This is the last thing in amino acid catabolism. Okay. If the amino acid catalytic enzymes will have some defect, what is the deficiency symptoms will happen? From here, from this entire table, you have to remember only 3 points. Maple syrup urine disease will be caused due to deficiency in the enzyme dehydrogenase complex, that is branched chain alpha keto acid dehydrogenase complex. If this enzyme is deficient, maple syrup urine disease will form. Phenylketonuria will be formed due to the deficiency of enzyme phenylalanine hydroxylase, and the most important thing, albinism will be formed, that is melanin synthesis will be defective if tyrosine 3 monoxygenase is defected. Okay. I hope this is clear for everyone. With this, we are completing the new fatty acid metabolism and amino acid metabolism. Any doubt is there? Any doubt is there? Kindly let me know any doubts are there. Kindly let me know any doubts are there. Don't take much pressure. Whatever I am saying, that one only you read. I know metabolism is huge topic. Any doubts? Kindly reply any doubts. If no doubts means we can have one break. Okay. I will see you after 5 minutes. Okay. I will see you all at after 5 minutes. Hello students, whether I'm audible? Can you let me know whether I'm audible? Yes, whether I'm audible. I hope I am audible now. Okay. Now we are going to see about nucleotide metabolism. Okay. How the nucleotides are synthesized. The key important point from nucleotide metabolism, we can expect only one question, but this is the deciding question. Okay. This is deciding question. Okay. So much of people will leave this nucleotide metabolism. Okay, so what are the important points? That are the things I am going to discuss today. Okay, so if you see the nucleotides, okay, adenine, guanine, thymine, cytosine, and uracil will be there. Okay, so the purines means adenine and guanine will be there. Okay, pyrimidines means cytosine, thymine, and uracil. These things you have to remember. Okay, uracil will be present in RNA molecule. In spite of uracil, thymine molecules will be present in DNA molecule. This is another important point. Okay. Then there are two different pathways of nucleotide biosynthesis are there. Okay. One is salvage pathway and second is de novo pathway. This you have to remember. First is de novo pathway. De novo pathway and second is salvage pathway. Okay. In de novo pathway, the nucleotides will be synthesized by utilizing the carbon and nitrogen source from different biomolecules. Okay. For example, carbon dioxide, aspartate, different amino acids will be utilized that will donate the carbon and nitrogen sources for synthesis of these nucleotides. In salvage pathway, what it will happen means if the nucleic acid is degraded, the base will be recycled. Okay. So the base will be recycled in salvage pathway. These are the 2 points you have to remember. Okay. Now we are going to see the synthesis of purine. Okay. How the purine molecule will be synthesized. The examples of purines are what? Adenine and guanine is the example of purine. Okay. Here we have just list out what are the different sources of carbon and nitrogen donors for purine nucleotide biosynthesis. Okay. The most important thing you have to remember is the commitment step for purine biosynthesis is what? Catalyzed by phosphoribosyl pyrophosphorate. Okay. The first commitment step of purine biosynthesis, it catalyzed by PRPP glutamine amidotransferase. Okay. PRPP glutamine amidotransferase. This is very important. Okay. You have to just remember the substrate PRPP and glutamine. From here, the amino group will be transferred. That's why we are saying PRPP glutamine amidotransferase. This is the first commitment step of purine biosynthesis. You don't want to remember all the step of purine biosynthesis. The most important thing you have to remember is in purine synthesis, the first purine molecule synthesis is IMP, inosine monophosphate. Okay, inosine monophosphate is the first purine molecule synthesis. Okay, from this IMP, how the GMP and AMP will be produced? This is important. Okay, so the first synthesis is IMP. From this IMP, GMP and AMP will be formed. Okay, AMP and GMP will be formed. This is very important. Okay. How this is synthesizing? This enzymatic reaction you just go through once. And the most important thing for the synthesis of AMP, GTP will be utilized. And for the synthesis of GMP, ATP will be utilized. So reciprocal regulations are there. If an excess of GMP is there, that regulation will be regulated by ATP molecules. I hope this is clear. Okay. So reciprocal regulation will be there. Okay. Now we are going to see the regulation, how the regulations of purine synthesis is there. I am going to say only the important points that are highly sufficient. Okay. If you see the regulation way, I told first commitment step is what? Glutamine PRPP amidotransferase. This step will be inhibited by AMP, GMP and IMP. This is very important. Okay. Apart from that, I told reciprocal regulation will be there. ATP will regulate the synthesis of GMP. GMP synthesis will be regulated by ATP. Sorry, ATP regulates the GMP synthesis GTP regulates AMP synthesis. These two are key important point you have to remember this. That's all that are important from biosynthesis of purines. Okay. Now we are going to see biosynthesis of pyrimidines. Okay. Pyrimidines means what it will come. Adenine, guanine is purine, so thymine, cytosine and uracil will come in the classes of pyrimidine nucleotide. Okay. Here you have to remember the first commitment step for purine by pyrimidine biosynthesis is ATCase. ATCase. Aspartate transcarboxylase. Okay. Aspartate transcarboxylase is the primarily enzyme. Transcarbamylase is the primary enzyme. That is the first commitment step for synthesis of pyrimidine nucleotide. Okay, you have to just remember this thing, that is highly sufficient. Okay, so aspartate will be there, that will bind with carbamyl phosphate, that to lead to formation of N-carbamyl phosphate. The first commitment step is catalyzed by aspartate transcarbamylase. Okay, and the most important thing, first pyrimidine molecule formed is OMP. Okay, OMP will be formed. From this OMP, UMP, GMP and other molecules will be formed. Okay, from this OMP, other molecules like UMP and CTP will be formed. Okay, you have to remember what is the first molecule formed in purine and pyrimidine biosynthesis. This is key important point. Okay, if you see the regulation, I told ATCase is the first enzyme or primary enzyme involved in pyrimidine biosynthesis. That, that will be positively regulated by ATP and negatively regulated by CTP. Okay, so ATP is the positive regulator. Okay, positive regulator. CTP is the negative regulator. Okay, negative regulator. These 2 points you have to remember. What are the examples of positive regulator and negative regulator? So, if an UMP or UMP is forming or AMP or GMP is forming, okay, how this is converting to GTP or how this is converting into GMP or GTP. These things are very important. Okay. How a monophosphate molecule is converting to diphosphate, how the diphosphate is converting to triphosphate. Here comes the different enzyme source. Okay. For example, adenylyl cyclase, adenyl, adenyl kinase. Okay. This is the enzymes that are specifically involved for conversion of AMP to ADP molecule. This is the most repeated question. Okay. Adenine kinase. Okay. That will be involved in conversion of AMP to ADP. Then nucleotide diphosphate kinase. Okay. This will be involved in conversion of NDKs to NTKs. Okay. This is very, very important. You just go through once. Okay. They can ask this time also. Okay. Now we are coming to see how the molecules, how the deoxy molecules will be produced. How the deoxy molecules will be produced. As we know, the DNA will have deoxy sugar. Correct. But RNA will have without deoxy sugars will be there. Okay. So RNR is the primary enzyme, RNR. That means ribonucleotide reductase. It is the primary enzyme that will be involved in conversion of nucleoside, that will be involved in conversion of NMP or NDP to deoxy NDP. So deoxy sugar will be formed by the action of enzyme RNR. You have to remember only these things. Okay, RNR is the primary enzyme involved in synthesis of deoxy NDPs. I hope this is clear. Okay, you don't want to read the enzymatic steps. This is highly sufficient. Okay. Now we are going to see how the deoxy TMP molecule, that means how the deoxy TMP will be formed from deoxy UMP. Okay. Here you have to remember one thing, the key difference between uracil and thymine molecule is what? There will be a presence of methyl group at fifth carbon. Okay. So, one methyl group will be there at the fifth carbon. Okay. How this methyl group is synthesizing that? we are going to see. Okay, from where this methyl group is coming. Okay, here comes the role of enzyme thymidylate synthase. Okay, the thymidylate synthase is the enzyme that is catalyzing the conversion of deoxy-UMP to deoxy-TMP. This methyl group will be donated by SAM, S-adenosyl methionine, that will be act as a donor for that methyl group. These are the two things you have to remember. Okay. Now we have completed how the nucleotides are synthesized. Okay. How the purines are synthesized, how the pyrimidines are synthesized. What are the important points? That thing only we have discussed in exam point of view. Okay. Now we are going to see degradation of purines. If purines are degraded, the question what they will ask means, if purines are degraded, what the product will form? If pyrimidine is degraded, what is the product will form? Okay. If purine is degraded, uric acids will be formed. If pyrimidine will be degraded means beta alanine and beta amino isobutyric acid will form. I hope this is clear. Okay. If purine is degraded, that will lead to the synthesis of what? Uric acid. If pyrimidine degradation will be happen means beta alanine and beta amino isobutyric acid will form. This everything I have discussed in our normal lecture only. Okay. This we have already seen deeply, very deeply we have discussed. Okay. Just we are revising now. Okay. Now we are going to see the salvage pathway of purine. I already told. Salvage pathway means what? The base recycle will be happened. Okay. So here comes the role of two different things. Okay. Two major enzymes are involved in salvage pathway of purine. Okay. First thing is adenosine phosphoribosylate transferase. Okay. Second thing is HGPRT. HGPRT. Hypoxanthine guanine phosphoribosyl transferase. These two enzymes are very, very important. Okay. These two enzymes are directly involved in base recycle for synthesis of purines and pyrimidine molecules. Okay. These two things you have to remember. And the most important thing, if HGPRT enzyme is deficient, that will lead to formation of Lesch-Nyhan syndrome. Okay. Lesch-Nyhan syndrome. This is another important and repeated question. Lesch-Nyhan syndrome will be formed if HGPRT gene is defected. Okay. Okay. Now we are going to see the inhibitors. What are the different inhibitors that can be used to inhibit the biosynthesis or synthesis of these nucleotides? Why we have to study about the inhibitors of these nucleotides? If you see cancer cells, we can use these drugs for treating cancer. Okay, so that because in cancer cell rapid multiplication will be there. So if we inhibit this nucleotide biosynthesis, the growth of the cells will be, growth of the tumors will be decreased. Okay, so there are different inhibitors are there. First is allopurinol. You have to remember what are the inhibitors and what is its enzyme. Okay. Allopurinol is the key enzyme that will act as an inhibitor for xanthine oxidase. So, inhibitor and its role. Okay, xanthine oxidase. Okay. Asacerin and acivicin. This will inhibit the first commitment step of purine biosynthesis. Okay. First commitment step of purine biosynthesis, that is glutamine aminotransferase enzyme will be inhibited. Okay. Then the third most important thing is aminopterin, okay, aminopterin, trimethoprim and methotrexate. These three are typical example for inhibiting the synthesis of deoxy-TMP molecules. Okay. So deoxy sugar synthesis will be inhibited by using this different class of inhibitors. Okay. This is very important. Okay. From here, sure they will ask one question. What are the inhibitors? From inhibitor point of view, sure they will ask one or two questions. Okay. Any doubt is there so far? Kindly let me know. If no other doubts means, Yes, if no other doubts means we will start carbohydrates metabolism. Okay. The last topic of today's lecture is carbohydrate metabolism. Already we have completed the respiration everything in yesterday class. Today we are going to see a just a glimpse. The remaining things we are going to see. Okay. We are going to see how the glycogen synthesis will be happen, how the glycogen degradation will happen. This is the two important thing we are going to discuss today. Okay. Then another most important thing, how the the deficiency of the glycogen synthesis is affecting the other functions. Okay, this is another thing we are going to discuss. At last we are going to see how starch and sucrose molecules are synthesized. Okay, how starch and sucrose molecules are synthesized. Okay, in glycogen synthesis, so glycogen is what? It's a storage molecule of glucose. Okay, that will have alpha 1 to 4 and alpha 1 to 6 linkage. So branching will be happened at alpha 1 6 linkage. Okay. Now the key important things only we are going to discuss. Glycogen synthesis. Glycogen synthesis, okay, will primarily happen in where liver cells will be act as a primary organ for synthesis of this glycogen molecule. The key enzyme involved in glycogen synthesis, that is another important thing. The key enzyme in glycogen synthesis is UDP glucose pyrophosphorylase. Okay, this is the key enzyme involved in glycogen synthesis. This is the thing they will ask in exam. Okay, UDP glucose pyrophosphorylase is the key enzyme involved in glycogen synthesis, and the source of glucose donor will be UDP glucose. Okay, UDP glucose is the source of glucose. This is also another important thing. If you see starch, ADP glucose will act as a source of glucose. Okay, this is will act as a glucose source. That means donor of glucose for synthesis of starch. This is the two important thing you have to remember. Okay. Then this is just, they have given the brief line of how the glycogen synthesis is happening. You have to remember here, the linkage is alpha 1 to 4 between glucose and glucose will be there. Okay. Then branching will be primarily happened at alpha 1 6 linkage. Okay. The branching will be happened at alpha 1 6 linkage catalyzed by glycogen phosphorylase, branching enzyme. Okay. These two things you have to remember. This is the same thing we have given here in linear pathway. Okay. UDP glucose will act as a donor for glucose. Enzyme will be there. Okay. Different catalyzing enzyme will be there. Branching enzyme will be there. Because of this, glycogen molecules will be synthesized. Okay. Now we are going to see how the glycogen synthesis is regulating. Here you have to remember only one thing. Okay. The glycogen synthesis will be regulated. The key enzyme is what? Glycogen synthase. If the glycogen synthase is phosphorylated, that will become inactive. Just I am writing here. Glycogen synthase enzyme is there. If the enzyme is phosphorylated, this will become inactive. Okay, inactive. If it is there in dephosphorylated form, that will be active. These are the two things you have to remember. Okay. Then now we are going to see glycogen degradation. So if glucose is needed in body, the glycogen has to be degraded to release the glucose molecule. Okay, glycogen should be degraded to release the glucose molecule. The primary enzyme involved in glycogen catabolism is glycogen phosphorylase. This is another important thing. Glycogen phosphorylase. Okay. Another most important thing you have to remember, the branch at alpha 1 6 will be removed by glycogen debranching enzyme. Okay. Glycogen debranching enzyme. Okay. Glycogen debranching enzyme. Okay, we are going to see only the important things. That's why I'm moving fast. Okay. Then another most important exam point of view, what they will ask means in glycogen catabolism, if you see, at last glucose 6 molecules will be formed. That glucose 6 will be converted into glucose, only free glucose molecule by the action of glucose 6 phosphatase enzyme. By the action of glucose 6 phosphatase enzyme, that will be present only in endoplasmic reticulum. This is another important point you have to remember. Okay. With this, we are completing the glycogen metabolism. Okay. Last thing, how the glycogen catabolism is regulating. Okay. The glycogen catabolism, the primary enzyme is what? Phosphorylase enzyme. If the phosphorylase enzyme is present in phosphorylated form, that means glycogen phosphorylase, if it is in phosphorylated form, it is active. Okay, so glycogen phosphorylase, if it is present in phosphorylated form, that will be active. If it is present in dephosphorylated form, it will be inactive. Okay, these are the two important things you have to remember. Okay, here I have just listed what are the glycogen catabolytic enzyme deficiency, and this is just you go through once. Okay, just you go through once. That is highly sufficient. Okay. Now we are going to see the last part of today's lecture. Okay, starch and sucrose biosynthesis. Okay. So the key point what they will ask, I already discussed. The donor for the glucose molecule for starch synthesis is what? ADP glucose molecule. The key enzyme involved in starch synthesis is ADP glucose pyrophosphorylase. ADP glucose pyrophosphorylase. These two things you have to remember. Apart from that, starch synthesis will be always happen in chloroplast. Okay. But sucrose synthesis will be happen in where? Cytoplasm. These two things you have to remember. Okay. These two things you have to remember. Now we are going to see how the starch molecules will be synthesized. This I already discussed, how it is synthesizing, how it is catabolizing, that also we are going to discuss here. So if you are taking starch-rich food, that will be acted by alpha amylase enzyme that will be present in the salivary of humans, salival glands. Because of this, glucose molecule will be released. Okay. But if you see in plants, the starch will be stored in grains. This starch will be hydrolyzed by starch phosphorylase enzyme. Starch phosphorylase enzyme. That is the enzyme primarily involved in degradation of starch molecules in grains. Okay, I hope this is clear for everyone. Okay. Then this is also similar with glycogen degradation only. Different deep branching enzymes will be there that will release the branch at alpha 1,6 linkage. Okay, I hope this is clear for everyone. Okay, starch catabolism, this we have already discussed. Simple only. Okay. Then last thing is sucrose synthesis. How the sucrose synthesis will happen? Okay, that we are going to discuss. Okay. So sucrose synthesis will be primarily happened in where? That I already discussed. Cytoplasm is the major site for sucrose synthesis. Okay. Then what will be the precursor for glucose molecules and fructose molecule? Okay. This is another important thing. UDP glucose will act as the glucose donor. Okay. UDP glucose will act as the sucrose donor. You just see here, UDP glucose is there. We know sucrose will have alpha beta 1 to 2 linkage of glucose and fructose. Okay. So UDP glucose will act as a glucose source. Then fructose will come from fructose 6 phosphate. These two will bind by the action of sucrose phosphate synthase. This is another important enzyme. Sucrose phosphate synthase will be there. The sucrose phosphate synthase will lead to the formation of sucrose. Okay. This is very, very important point. Okay. With this, we are completing our metabolism of carbohydrates. I hope there is no doubt in this. If any doubt is there, kindly let me know. I will teach. Okay. With this, we are completing the metabolism of carbohydrate, proteins, lipids, and amino acids. Okay. Any doubt is there? I think no doubts are there. Okay. I will see you all in next lecture. Okay. Tomorrow we will see the remaining things. Huh? Thank you all.